All right, it is 8 a.m. So let's get started for today. So to begin with, just some reminders that next week, Thursday, so the 14th, be test 2. All the PowerPoints that are going to be on test 2 are already up on the Blackboard site. So that should all be ready to go. And it's going to be the same idea as in test 1. We'll have an hour of class, and then we'll jump right into test 2 next week, Thursday. So make sure you are studying for that. All right, so let's just go over what we talked about on Monday. And we were looking at hemoglobin and myoglobin. That's the last topic we left off on. And we were looking, the main idea here is that we were looking at how proteins and ligands bind each other. So we were looking at binding constants. So we're looking at this graph where we have fractional saturation. Remember, that's the amount of protein that is bound to a ligand versus the amount of that ligand. And we also looked at cooperative binding for hemoglobin. And hemoglobin has 4 hemes. So that means it can bind 4 oxygens at a time. And the thing about hemoglobin is that if one oxygen binds at one heme, the other hemes want to bind oxygen as well. Their binding affinity goes up. It makes them more likely to want to bind those oxygens. And we can tell that on a graph of fractional saturation versus pressure by this sigmoidal curve, right there. So the curve kind of looks like an S. That S shape is telling you positive cooperativity. If you look at myoglobin, there's no S shape to that curve. It just goes straight up, and then it flattens out as you are completely saturated with oxygen. And then the very last thing we did was look at data sets and look at them on a plot and determine which one is more likely to be cooperative binding. All right, so the next thing we're going to talk about is the Hill plot. And what a Hill plot is it tells you if a protein has cooperative binding or not. And so what you do is that you plot fractional saturation divided by 1 minus fractional saturation, which is a weird idea, but that's what the y-axis is. And the x-axis is the ligand concentration you're interested in. For hemoglobin and myoglobin, we're binding oxygen, so that's why we have pressure oxygen on the x-axis. Then, after you plot your data, you measure the slope of the line, all right? So myoglobin, if we look at myoglobin's data, which is this straight line, right? The slope is 1. And that means the Hill coefficient for myoglobin is 1. And if your Hill coefficient is 1, that means you do not have any cooperativity. That is, the binding of one ligand does not affect any other binding sites. And for myoglobin, this makes perfect sense. Myoglobin only has one binding site. Therefore, if I'm a myoglobin and I bind oxygen, I have nothing to influence. So to have cooperativity, you have to be able to bind multiple ligands in the same protein. Myoglobin can't do that, so it has a Hill coefficient of 1. But let's look at hemoglobin. And hemoglobin, to begin with, at the lower asymptote here, has a Hill coefficient of 1 as well. So at very low concentrations of oxygen, hemoglobin won't bind oxygen. So it looks like there's no cooperativity there. However, as the oxygen concentration increases, our line starts to go up. And now the slope is no longer 1, but it is roughly 3. What that tells us is that if your slope is greater than 1, you have positive cooperativity. That is, you bind one oxygen, the other sites want to bind oxygen even stronger. And then at the very top, when we're saturated again, that is, everything is bound, we're going to flatten off and no longer show that cooperativity because we are already bound. All 4 sites are bound. So when looking at a Hill coefficient, if you are not just a perfectly straight line, you take the slope of where your line is starting to curve. Here we are seeing it is 3. Now, it is also possible that you can have what's called negative cooperativity. And negative cooperativity means if you bind one ligand, your other binding sites do not want to bind that ligand. So you lower the chance of the other binding sites binding that ligand. So if hemoglobin had negative cooperativity, when one oxygen binds, it is harder for the other sites to bind oxygen as well. That is called negative cooperativity. And you would have a Hill coefficient of less than 1. So those are your 3 options. You can have a Hill coefficient greater than 1, less than 1, or equal to 1. And don't worry too much about the equations here. They're for completeness. I'm not going to ask you to do any math with these equations. But I do have a question, and I do have a poll for that question to start off here. And it's actually a question from your reading guide. And it seems every year since I started the reading guide, somebody always asks me the answer for this question. So let's see a poll to determine what people answer for this. I'm wondering, is it possible that you can have a Hill coefficient, which gives the variable n? Can you have a Hill coefficient of 0? So this is simple yes or no. So please answer that, and we'll talk about it in a minute. We'll do about 15, 20 more seconds. So please answer now if you haven't yet. All right, and let me just quickly download their results here. Okay, so I have that downloaded and saved for future reference. All right, so let's remember what slope means. So hopefully back in your middle school, maybe high school math classes, you learned that slope of a line is equal rise over run. At least that's how I was taught. The change in the y divided by the change in the x. So if this is equal to 0, we have two options. Either our line looks like this, that would give you a slope of 0, or it looks like this. That would give you a slope of 0 as well. The first plot on the left does not make sense for our axis, because basically what we're saying is that the ligand concentration would never change, yet we could be bound from 0 to 100%. And in this experiment, what you're doing is you're changing the ligand concentration. So the only possible way for a slope to have 0 is this. And technically, it is possible. So if you want to go to the technical sense, yes, you could have a slope of 0, or sorry, a Hill coefficient of 0. But let's understand what that means, right? So if I had a slope of 0, then that means my line would look like either this, oops, and let me get a different color instead of black. Or it would look like this. Those are pretty much your only two possibilities. It would either be all 0 or all 100. And what that tells us, if it's down here, that means no matter how much ligand you have, your protein would never bind that ligand, which isn't that interesting to us. Like nobody would actually say, oh, that protein doesn't bind that ligand. It has a Hill coefficient of 0. You wouldn't bother with that sentence, right? You would just say, oh, that protein doesn't bind that ligand. Or it's no matter what ligand concentration you're at, you're always bound, which again, nobody would care about that because that's no longer a ligand. That's called a prosthetic group. It's always there. It's probably like covalently bound to your ligand. So my answer for this is technically yes, but practically no. You could have an N of 0, but that's not useful. Nobody would ever like comment on that, if that makes sense. So I guess I would say both yes and no. Yes in the technical sense, no in the practical sense. But yeah, any questions about the Hill plot or Hill coefficients or anything up to this point at all? All right. Let's move on then. If you are typing on questions, feel free to continue to do that, and I'll address it when I see it pop up. We do have 2 more questions on here, and they all have to do about math. So we're actually going to skip these for the sake of time. So we're going to skip question 6 and 7. What that means is that I'm not going to ask you to use this equation on the test. So when looking over your notes, I won't ask you something like questions 6 and 7 on the test, which is just basically math-based. But do know what Hill coefficients mean. Do know what the possible values of your Hill coefficients are, and what that tells you about cooperativity. And with that, that ends the little bit of the PowerPoint we had left over from Monday. So let me pop up the PowerPoint I had for today. Stop sharing that. Share. 1 second. Still in editing mode. Share this. All right. And let me get my Zoom controls back up. Perfect. Oh, okay. Thanks for letting me know, as that is one way I do know if people here are here or not. All right. So we've talked about hemoglobin having positive cooperativity, but we didn't really go into the specifics of like how that happens. How does a protein know? And let's draw my little crappy hemoglobin cartoon again. So remember, hemoglobin has 4 subunits. Each subunit, there's a heme, which I'm putting that X there. And what we're saying is that oxygen binds at 1 heme. All 3 other hemes know that happened, and now they are more likely to bind oxygen. We're going to go into how that actually works, because proteins can't talk to each other. So how can one thing happen in this subunit and directly affect the behavior of 3 other subunits? And to talk about that, we're going to bring in 2 different states your hemoglobin can be in. Either the tense state or the relaxed state. So T state or R state. And tense and relaxed are actual terms that you'll see in biochemistry, molecular biology a lot when it comes to proteins and ligands. And they really just mean, is a ligand bound or is it not bound, for the most part. And for hemoglobin, if you're in the tense state, that means you do not have oxygen bound. If you're in the relaxed state, you do have it bound. So tense, no oxygen, relax, oxygen. Now, it's not as simple as no oxygen or oxygen, as we're going to see in a minute, but that's a good way to begin to think about it. And the way I really memorize this is that I did a stupid little skit in my head. Like if you are in a room without oxygen, you're going to be very tense. You're going to be like, oh, I need oxygen very, very bad. And you're going to be all tensed up. Well, if you have oxygen, you're relaxed. Nothing in the world can bother you. You're just feeling good. You're nice and relaxed. So tense state and relaxed state. And hemoglobin's nice because when we color code that blue and red, you can think about that as blood as well, right? Veins, blue blood, no oxygen. Arteries, red blood, and it's been oxygenated. All right, so that's the 2 states hemoglobin can be in. Now let's look at the structure of these 2 states. And we're going to begin with the blue. So let me just outline it. So we're really just, ignore the red for right now. We're just focused in on the blue. And then our F helix up here. And if we're in the tense state, we don't have oxygen yet. So imagine that oxygen is gone. And so we're just looking at the heme ring without oxygen there. And right now, iron, the iron molecule in our heme ring, is out of the plane of our porphyrin ring. the porphyrin ring, if you remember, is just like a bunch of histidines. They're not histidines, they have been modified, but they look like histidines. So it's like a net. And in the middle of this net is your iron. And when you don't have oxygen bound, when you're in the 10th state, the iron is a little bit popped out of that net. And remember, if you remember from Monday, I didn't really talk about it a lot, but there is a histidine residue, his F8, that is always going to be interacting with your iron, both in the 10th state and in the relaxed state. All right, so that's when we don't have oxygen. That's what your heme looks like. Then when you go into the, there we go. Whoops. Then when you go into the relaxed state, a lot of things happen. So oxygen comes in. When oxygen comes in, your iron molecule, which is positively charged, moves 0.6 angstroms towards that oxygen, which is 0.06 nanometers. So it doesn't move far, but it moves a little bit towards that oxygen. When that iron moves down, the ring, the porphyrin ring, also shifts up. And now the iron molecule is perfectly in line with your porphyrin ring. They're all in the same plane. Now, with the iron moving down, this histidine that was attached to this iron or is interacting with that iron also gets pulled down by roughly 0.6 angstroms. This histidine is connected to the rest of your hemoglobin subunit. And in the big part of the protein that's right next to your heme is called Helix F. And the whole protein shifts in response to that iron movement. So oxygen comes in, iron moves towards it. Iron is connected to the rest of the hemoglobin, so the rest of the hemoglobin moves with that iron. And what happens there, if you remember that whoops, that's way too big of a circle. Let me undo that. Hemoglobin is interacting at all the interfaces, right? They're all touching, even though I didn't quite draw it like that. They're all interacting, all four subunits. And so when one subunit binds to oxygen, that whole hemoglobin changes conformation. And what I'm showing here is that the interfaces, you change your interactions. So in the T state, we're looking at an interface between a beta and an alpha subunit. You can see we have an aspartic acid on the beta subunit interacting with a tyrosine on the alpha subunit. When we go to the R state, this whole beta subunit will have shifted, and now we're making a hydrogen bond between asparagine and aspartic acid, and this aspartic acid is no longer interacting with anything. And at the termini as well, so here we're looking at beta with another alpha, and actually multiple alphas. We have this hydrogen bond network in the T state. That's what all these dots are. So we have a bunch of hydrogen bonds. We have a bunch of salt bridges in the T state. In the R state, we are breaking a lot of these, and we are actually changing hydrogen bond partners. This change in interactions is how hemoglobin talks. This is a signal to the other three subunits that, oh, one subunit just went into the T state, or sorry, just went into the R state. This means oxygen's available. So what the other subunits do in response to that is that they also adopt the R state. They make it easier for oxygen to bind because that iron is just waiting in the pocket where oxygen binds. It's inviting that oxygen in. So this is how we have positive cooperativity, where oxygen binds to one subunit, iron follows that oxygen, changes the conformation of that one hemoglobin. The change in conformation changes the interaction at all the interfaces. This change in interaction causes these other hemes to shift from T to R as well, making it easier for oxygen to bind in those hemes because we already have a charged iron waiting at the oxygen binding site. That, in a nutshell, is how positive cooperativity works in hemoglobin, all due to protein movement. And since this is all due to interactions between charged molecules, no ATP is needed for these movements. This happens energy-free. It's all spontaneous upon oxygen binding. Right, so is there any questions about that idea? That's actually quite a lot of information on one slide, but any questions about this positive cooperativity or T/R idea? The tense and relaxed state. All right. To shift, so question, are we going to look back at how they shift back to the T state? It's 100% reverse, right? If you want to go back to the T state, the arrow goes this way, goes that way. It's just and the iron moves back out of the ring. So if you're in the R state and you're going to the T state, oxygen leaves, it's delivered. If there's no oxygen there, the iron is just going to be like, okay, well, I have no reason to be in this pocket anymore. I'm going to go back. When it goes back, it shifts everything back as well. All the other interactions change and everything, and all the other subunits are saying, oh, one heme just released this oxygen. That means, and we're going to look at, well, we already looked at it, but under low oxygen concentrations, heme just releases oxygen. That's what it does. And that's good because it's delivering oxygen to the body. So under low oxygen pressures, heme will just naturally release that oxygen. And it's just the reverse of what we just said. It works in 100% the reverse manner. There's nothing different from going from T to R and then R to T. How does it let go of his charge relationship? It's all due to diffusion, right? So I don't think I have that plot. That was on the last PowerPoint. But if you remember, if we're looking at fractional saturation versus pressure of oxygen, hemoglobin more or less looks like that. And under conditions of low oxygen, if hemoglobin finds itself in the area of low oxygen concentration, we are now talking about entropy again, right? Delta G equals delta H minus T delta S. For oxygen to be bound in hemoglobin, that's very entropically unfavored because oxygen can no longer diffuse. It can no longer spin around. It's stuck in one position. It has good interactions, so that's good, but the entropy is bad. If you have a lot of oxygen, though, if you're at very high pressure, the entropy penalty for oxygen isn't that bad, though, because all the other molecules that are not bound, and remember, this is a very hard concept to think about, because you really have to think of the universe when you're talking about this. And so if you're in a localized area of high oxygen, all the other oxygen molecules, they're free entropically. They can move, they can diffuse, they can spin, they can stretch, they can do whatever they want. So if a few molecules of oxygen get bound, that's okay. The universe is happy with that because most of the oxygens are still free. You go to an area of low oxygen concentration, though, that entropy penalty of being bound outweighs the interactions between the heme and the oxygen as well. So the delta H is not enough to make up for oxygen being bound if there's not a lot of oxygen present. And so oxygen will be released to be entropically free, right? That makes a spontaneous process now. So that's why at high concentrations, we bind oxygen. At low concentrations, we don't. It all comes back to the relationship between entropy and enthalpy. When are those conditions favored to bind? And when are those conditions favored for oxygen to be free? Well, and this is something I stretch in Gen Chem 1, or I think Gen Chem 2 is when we actually look at this. This equation, delta G equals delta H minus T delta S, is like one of the most important equations in all of science, all of at least chemistry, biochemistry, even physics. This simple equation more or less explains everything, if you can actually boil it down to that. All right. So this page is just like a bullet point of everything we've talked about so far. So this is a good idea for when you are going back to study, remember tests in 1 week. Everything I just said and actually laid out. So I'm not going to read it here as I just said it, but this explains the previous page. So let's do some critical thinking now. So here's a question for everybody. Myoglobin and hemoglobin look more or less the same. In the bottom here, I'm showing a picture of myoglobin, and I'm showing one subunit of hemoglobin, a beta subunit. And they more or less look the same, right? They both have like a V here with another helix below the V. In the middle of this V, you have a heme. If you compare the amino acid sequence between myoglobin and hemoglobin, it more or less, again, looks the same. There are some differences, but for the most part, they have very similar amino acid sequences. And remember what I said, I believe, last week, when comparing proteins and you want to know if they have similar function, you actually don't care about their sequence. You care what they look like. If they look similar, they're probably going to do the same thing. So the question I have for everybody here is that if I took out the beta subunit of hemoglobin and I put in myoglobin, do you think you would still have cooperativity? Do you think that this myoglobin would have the same function as the hemoglobin and work like with no problem? So it would be a one-to-one swap. and why would you answer the way you did? So I'm going to give people a little bit of time to think that over. And when you have a thought on that, feel free to pop it in the chat. Say yes or no, and just give me a very brief explanation as to why you think this is. This is good practice for any short answer questions you might have on the test. So think that over and put in an answer when you have one. So I'll give you just a little bit of time to think this one over. So we have a few people chiming in. So I'm just give people a little bit more time. If you haven't have an idea yet, please do so. What are you thinking about this based on everything we have talked about in biochemistry so far? All right, so let's look at some of these responses. No, because the surface charge of myoglobin would not permit it to bind properly with the hemoglobin subunits. Yes, because they are structurally similar, question mark. I don't think so, because even if they are similar, they are not the same. They'll still have different sequences, so I don't think it would work. I believe so, but not 100% sure. Would it be no because of the surface charge of the myoglobin? Then I'm leaning towards yes because of the idea of peptide substitution, how the two are similar, but also no because of structural differences. So thank you all very much for answering that. All very good ideas, all using the knowledge you have learned previously based on proteins and peptides. Another one, I would say no, because myoglobin has no cooperating, meaning that higher oxygen concentrations of oxygen, it won't let go of the oxygen. This in turn would mean that the other subunits of hemoglobin wouldn't know when to let go of oxygen or not. All right, so the answer is no. You could not substitute a myoglobin for a hemoglobin. And the reason for this is that even though they look the same, the actual, and people were correct in saying that the surface charges of the myoglobin matter. They do matter a lot when it comes to hemoglobin. So even though they look the same and they have more or less the same function, their charges, the exact amino acids, do matter quite a bit, because if we go back to the previous slide, you see that the amino acids that are interacting between the subunits are very critical. These amino acids cannot change. In hemoglobins, these would be invariant amino acids because they are very important for communicating information between the different subunits. And myoglobin does not have those same exact subunits. So I think it would be possible to actually put myoglobin in where hemoglobin would be, because it's structurally they look the same, they have about the same size, but you would not have any cooperativity. Myoglobin and hemoglobin could not communicate because they don't have the same exact amino acids at where the different subunits interact with each other. So what would happen is that the three remaining hemoglobin subunits would probably still have cooperativity with each other, but myoglobin would more or less be blind and deaf when it comes to the other three subunits. They don't know what's going on. So they're just going to act like a myoglobin. Oh, I'm just going to bind oxygen and hold it very tightly because I don't know what cooperativity is, right? So that's what I would think of that answer. Any questions about that idea or any clarification on that idea? All right. Now we can go on. Now, these questions deal with, do you understand what the R in T state means? And what does P50 mean? Remember, P50 is the amount of oxygen you need to be 50% bound and unbound. And in hemoglobin, we have a lot of different mutants that can cause different diseases. Sickle cell anemia being the most famous one. So here I have two questions. So I'd like you to think about this and answer when you think you have a solution. So in our first variant, Yakima, we have a cysteine being mutated to aspartic acid at position 99. That's what C99 and D99 mean. It's just saying cysteine position 99 mutated to aspartic acid position 99. When you do this, you stabilize the R state. So is P50 higher or lower compared to normal hemoglobin? And then hemoglobin variant Kansas, you see they're named after where they were first discovered. Asparagine 102 is mutated to threonine 102. We now stabilize the T state. So is P50 higher or lower than normal hemoglobin if you stabilize the T state? So go back, review R and T state, see if you have this correctly in your head, what these mean, and then relate that to P50. So again, I'm gonna give people a couple minutes, think about this, and when you have an answer, let me know, and then we'll come back and discuss it after people had some thought about this. Remember, P50 is oxygen concentration or pressure oxygen, 50% bound, 50% unbound. So what are people thinking? Let me know. Let me do some drawings just so we remember, and I'm going to color code it. So if you're in the R state, so that's iron, and these are our porphyrin rings, which I'm just going to write P instead of porphyrin. And that's our oxygen binding site. Oxygen is in the plane of the rings, ready to bind that oxygen. And if you're in the tense state, the iron is not in the plane of the ring. So there's actually a greater distance between the oxygen binding site and your charged iron. So I'll give like one more minute for people to think this over and see what you think. All right, let's discuss this. Seems to be some confusion on exactly what will happen. That's fine. That's why we have class to understand these ideas. And I just want to reiterate, in case you're thinking, why should we care about hemoglobin so much? Remember, this is just our model system. And the things we learned here, you can apply it to almost any protein binding to ligand system. All right, let's talk about Yakima. So I stabilized the R state. In the R state, my iron is in my porphyrin ring, which is right next to the binding site. So the R state wants to bind oxygen. Or I guess, instead of wants, because proteins don't want to do anything, more likely to bind oxygen. That's the more scientific way to put it. Since you're more likely to bind oxygen if you're in the R state, the pressure at which you are half bound and half unbound is lower than normal hemoglobin, because in Yakima, you're stuck in the R state, right? You're always going to want to bind oxygen. So it's going to be easier for oxygen to bind. Therefore, it takes less oxygen to be 50% bound than normal hemoglobin, who will be in the T state and makes it harder to bind sometimes. And then once you're bound, you go through that T to R transition. But in the R state, but in Yakima, there is no transition. You never go back to the T state. You're always in R, which makes it easier to bind. It makes it harder to release that oxygen because you'll never go in the T state to release it, but it's very easy to bind that oxygen. While in Kansas, you're in the T state always. You can never go in the R state. This means it is very hard to bind oxygen. That iron is never going to come to the oxygen binding site in all your subunits. It's always going to stick in the T site or the T state. So you need more oxygen to be bound 50% of the time for your hemoglobin. So the P50 raises. So for example, I don't know the exact numbers off the top of my head, but let's say for normal hemoglobin, so that's going to be NHB, like the P50, let's just say it's 20 torr, right? It takes 20 torr of oxygen. And if you don't know what torr is, it's just a pressure unit. And again, that 20 is actually not the real number, but this is just an example. So for normal hemoglobin, let's say it takes 20 torr of oxygen to be half unbound, half unbound, half bound, half unbound. So 50% of your hemoglobin have oxygen, 50% do not. Well, in Yakima, if I'm always in the R state, I can bind oxygen super easily. So let's say that only takes 10 torr to be 50% bound and 50% unbound. While Kansas, I'm always in the R state, or sorry, the T state. If I'm always in the T state, it makes it harder to bind. So let's say that's now 30 torr to be half bound, half unbound. However, both of these mutations make it harder to deliver oxygen to the body. For Yakima, when your hemoglobin goes to your tissues, it won't want to release that oxygen because it's not going in the T state. For Kansas, it's hard for that oxygen to be absorbed in the lungs in the first place because you're always in the T state. So you're delivering less of it to your tissues. So now that you have that example and you thought about the R and T state and you thought about its relation to P50, is there any questions about any of the terms or any of the ideas we have covered so far for hemoglobin? All right. Hopefully that cleared things up for you. If not, please do let me know, or have a meeting with me and we can talk about it some more. Otherwise, let's move on to our next slide. And we're going to talk about the Bohr effect. Now, in hemoglobin, there are many factors that can influence whether you're in the T state or the R state. And one of those factors is pH. And when you're in acidic conditions, that is, pH is low, it is harder for hemoglobin to bind oxygen. So that's the first bullet point there. Under acidic conditions, hemoglobin binds oxygen weaker. We can see that on the plot to the left. And again, let's look at P50. So at a pH of 7.6, P50 is roughly 22 torr. At 7.4, it is like 28 torr. 7.2, it's like 35 torr. So as you get more and more acidic, you're less likely to bind oxygen to hemoglobin. And hemoglobin, when it binds oxygen, it actually makes the solution it's in more acidic. Because for every oxygen that's bound, roughly a 0.6 of a proton is released. You might be asking yourself, how can a fraction of a hydrogen atom be released? What this 0.6 should mean to you is that when oxygen binds hemoglobin, there's a 60% chance that a proton will be released, right? 60% of the time, hemoglobin will release a proton, 40% of the time it doesn't, right? So what this means is that acidic conditions, low pH, favor the T state. When you're in conditions of low pHs, hemoglobin wants to be in the T state because of the different interactions that happen. And we're not going to actually look at that in depth. I believe your book does go into that. But just keep in mind, low pHs, you're going to favor the T state. When you're in the T state, you don't want to bind oxygen. And this is actually a very good thing, right? And that is shown down here in this chart. And we're going to go over this chart to show you why the Bohr effect, and remember the Bohr effect, and I'll probably have a question right in the test that says, like, what is the Bohr effect? Or just know that Bohr effect is acidic conditions release oxygen. Keep that in mind. So we're going to look at why this is good for us. And we're going to start right here in this circle. So we're in the lung, and you just took a breath of air. So when you take a breath of air, the oxygen pressure inside your lung is super high. It's 100 torr, right? At 100 torr, basically all hemoglobin that's in your lungs will bind oxygen because the pressure is just so high. So that's what's being shown here. Hemoglobin binds oxygen at the lung. Then it will go and travel throughout your bloodstream. And what happens is that hemoglobin will release oxygen at the cell surface, and myoglobin will pick that up and deliver it to the cell. So myoglobin will be in the cell, and it will take the oxygen from hemoglobin and deliver it. And it does this because myoglobin binds oxygen much more strongly than hemoglobin does. So myoglobin really wants oxygen more than hemoglobin. Now, that's what happens. Now let's imagine that you're exercising. And so we're looking at the muscle cells. And when you're doing respiration, you're creating carbon dioxide and water. So you're breaking down glucose, making carbon dioxide. And we have to get that carbon dioxide out of your system. However, carbon dioxide is a nonpolar gas. And so it cannot dissolve in your blood. That's a big problem. It would be a gas bubble in your blood, which can cause a lot of different conditions. One of them being the bends. If you know that from scuba diving, you can get gas as CO2, which can cause like strokes if it goes to your brain, or your heart will cause big problems. So since we don't want a gas in our blood, what we do with carbon dioxide is through an enzyme called carbonic anhydrase, we change it into carbonic acid. Carbonic acid is soluble. Now, this is where it gets interesting, at least to me. Carbonic acid, when making carbonic acid, you release a proton. So at the blood around your muscles, pH is really low because you're making a lot of carbon dioxide. Therefore, you're making a lot of carbonic acid and releasing a lot of protons. So the blood right around your muscles is very low pH. So hemoglobin will come around your muscle, and this proton will bind to hemoglobin. And the Bohr effect, when you're in low pH, you don't want to be bound to oxygen if you're hemoglobin. So in this area of low pH, hemoglobin releases oxygen, which is perfect because the muscle is working very hard. It needs oxygen to continue aerobic respiration. So imagine you're like doing a marathon, your leg muscles constantly need glucose. So the blood around your muscles is going to be low pH because of this carbon dioxide, but they're going to continuously get a new supply of oxygen because that low pH is stimulating hemoglobin to release oxygen to your cell. All right. Then with this proton will actually bind to hemoglobin, and hemoglobin does not have any oxygen, so it'll go back to the lungs to pick up new oxygen. At the lungs, you also have carbonic anhydrase, and this will change your carbonic acid back to carbon dioxide at the lungs, and you breathe out carbon dioxide, and that's how you get rid of carbon dioxide from your muscles, and at the same time deliver oxygen to your muscles. So thanks to the Bohr effect, when you're actively using your muscles, you also increase the amount of oxygen that is being delivered to your muscles. At the same time, you are getting rid of the carbon dioxide waste product, thanks in part to hemoglobin carrying that proton to the lungs to help reform carbon dioxide, and you breathe it out. Just one last thing. This carbonic acid is actually a buffer. It can be H2CO3 neutral or HCO3 minus. This is what keeps the pH of your blood around like 7.2 is the amount of carbonic acid with this base. So this is always in like some kind of equilibrium. And this prevents your blood from spiking too acidic or too basic. Yeah, and it's all part of the system as well. I think this is a very cool idea, or phenomenon rather, that just shows you how, once we learn the basics of biochemistry on the very tiny protein front, we can then take a step back and look at something like this and understand, oh, that's how the body works. It's to me, that's very awesome. But yeah, any questions about the Bohr effect or that chart I just talked about, showing how the Bohr effect is responsible for your muscles, your active muscles, gaining a continuous supply of oxygen. Right? If that is crystal clear for people, then we can move on. If it's not crystal clear, be sure to ask me any questions that come to mind. Or if you just want a quick refresher, I can also do that too. Now, that's the effect of pH on hemoglobin Bohr effect. The next molecule we're going to talk about is called BPG, 2,3-bisphosphoglycerate. Now, the image right here in the center of this protein, this is hemoglobin. So alpha, beta, alpha, beta. And at the center of hemoglobin, you have a lot of positive charges. And in the center there usually sits a molecule called BPG. And what BPG does is that it stabilizes the T state, right? It makes it hard for hemoglobin to bind oxygen, which again is shown here in the P50s. So if we just have hemoglobin by itself and no other molecule, the P50 is about 11 torr. Once you add BPG, the P50 is like 18 torr. So BPG stabilizes T state, which means it's harder for hemoglobin to uptake oxygen. And you might be asking yourself, why would we want a molecule that makes it hard for hemoglobin to bind oxygen? Well, there's a couple reasons for that. The first reason is shown on the chart on the bottom right. All right, so what we're looking at here is basically the pressure of oxygen in your veins. So that's this right here. And we're looking at the pressure of oxygen in your arteries at 4,500 meters above sea level and at sea level. So let's just focus on sea level right here. And the black line is the normal amount of BPG you have in your body. And the red line is if you have high levels of BPG. So we're focusing on the black line and we're focusing at sea level. So in your arteries to your veins, the amount of hemoglobin that's bound to oxygen changes by roughly 38%. What this means is that when hemoglobin leaves your lungs and it goes to a vein, 38% of the time, that hemoglobin will release an oxygen to that tissue because of the differences in pressure. So that's under normal conditions. 38% of hemoglobin will release their oxygen. If, however, you go 4,500 meters above sea level, the pressure of oxygen in your arteries is quite less. It goes from 100 torr to roughly 55 torr. This means that when you're above sea level, only 30% of your hemoglobin is delivering oxygen. So you have lost 8% of your oxygen delivering capabilities. If you've ever been that high above sea level, you would know that because you feel short of breath. You run, if you exercise or run, you find it's very hard to recover your breath. You always feel tired. That's because your body is actually delivering less oxygen to your cells. However, what will happen if you live at those high altitudes for a couple of weeks? You will increase the amount of BPG in your body. Your body will naturally start making more BPG. So that's this red line. And so let's say you lived in Denver for like 2 weeks. You have increased BPG. And then following the red line, after that increase of BPG, you're now delivering 37% of your oxygen, which is the same level that you were at when you were at sea level. So you got used to the altitude, you got used to low oxygen because your body increases the amount of BPG. That's why people go and train in the mountains, right? If you train at high altitudes, your body will create more BPG. And then when you go back to lower altitudes, you are really, really good at delivering oxygen, right? You might deliver like 45% of the amount of oxygen instead of 38%, which means your body can do oxidative phosphorylation at a much higher rate than if you just stayed at sea level. So that's how you can become like a super athlete after training at high altitudes for a couple of months. Eventually, though, your body would go back to normal rates. This is also good for pregnant mothers. So fetuses, they have fetal hemoglobin. They don't have regular hemoglobin like we do. They have a special type of hemoglobin that they only get when you're a fetus. And fetal hemoglobin does not bind BPG, which means they're like always going to be in the R state. So when the mother's blood, the mother's hemoglobin comes near the fetal hemoglobin, which is like always in the R state, will steal that oxygen from the mother's hemoglobin, which is a good thing because you don't want your fetus to be oxygen deprived. So it will just say, thank you for that oxygen. I want it more than you do. I will take it. It is now mine. So that is two reasons why BPG is actually very important for us. And what happens normally when you go to the R state, so if one subunit goes to the R state because it binds oxygen, then BPG is actually ejected from hemoglobin, and now all the other subunits can go to the R state. But while it's in there, it's going to stabilize the T state. And that's the effect of BPG on the oxygen binding capabilities of hemoglobin. So any questions about that idea or any clarifications or anything at all? All right, no questions? Go on then. And so here's a question I have for everybody here. Again, to just make sure we have all these concepts down. So another hemoglobin variant, Rainier, a tyrosine at position 145 in the B subunit. That's what tyro 145B means. Position 145B subunit, is replaced by cysteine. So you have a mutation. When that happens, you form a disulfide bond with a cysteine that's right near it. If this happens, you can no longer be in the T state. You're going to be stuck in the R state in Rainier. Therefore, I want you to tell me in Rainier, how does the oxygen affinity, the Bohr effect, and the Hill constant change compared to normal hemoglobin? So again, this is a good way to check your understanding of all these ideas that we've been talking about today. When you have thoughts on that, please tell me what they are in chat, and we will meet back to discuss this in a few minutes. So look over your notes, think about this. When you have some kind of conclusion, even if you're not feeling confident about your conclusion, just send me a private message. I won't put any names to it, but yeah, just tell me what you're thinking. So as a way for me to gauge our understanding. But yeah, take a couple minutes to do that. I'll be back to discuss this. So what are we thinking here? Let me know. Again, take what we've learned about the T state. You don't want to bind oxygen, and tell me you're in the T state, what happens to oxygen affinity? What happens to the Bohr effect? And what is that Hill constant? Remember, Hill constant has to do with cooperativity. Oh, sorry, yeah. It prevents the stabilization of the T state. My bad. So it will always be in the R state. So I'll give people like 1 more minute. So even if it's like, even if you're guessing or not sure, let me know, because this is answering the polls and putting stuff in the chat. It's really how I'm gauging involvement with this class, right? And that is part of your grade. The person who put B in chat, this is not a multiple choice question, by the way. All right. So let's take a look at this. Seems like we're having a little bit of difficulties with these concepts, which is good to know that, so we can address this. All right, so Rainier. Let me get my pen out. Oh no, Rainier prevents formation of the T state, so it's always in the R state. The R state wants to bind oxygen. So if you're always in the R state, well, oxygen affinity increases, right? Because I want to bind oxygen. So that goes up. The Bohr effect. So the Bohr effect says, under conditions of low pH, the T state is favored. However, I have a mutation that does not allow the formation of the T state. So the Bohr effect is the overall effect is lowered or non-existent, right? Because the Bohr effect only works by forcing hemoglobin to be in the T state at low pH. If I can never be in the T state because I have a mutation, well, it doesn't matter what the pH is, you're not going to change me. So the Bohr effect is non-existent. It's the effect of it is lowered. The Hill constant, right? So the Hill constant is a measure of cooperativity. And what the Hill constant is saying, or the reason why, let's just review the Hill constant, right? So Hill constant can have like 3 values. N is greater than 1, positive cooperativity. N is lower than 1, negative cooperativity. N equals 1, no cooperativity. And we talked about that hemoglobin has cooperativity because upon binding oxygen, a subunit goes from the T to the R state, and this is transmitted to the other 3 subunits. So the other 3 subunits will go to the R state as well, making them more likely to bind oxygen. Well, if I'm always in the R state, I don't have a transition. There's nothing to transition to. I'm just in one form. Therefore, the Hill constant is lower compared to normal hemoglobin for Rainier, because N would be 1, right? Cooperativity only happens in hemoglobin because it has these 2 states. It has T and R. And the ability to communicate to the other subunits to what state you're in and influence them through that transition. So if I'm in the R state and I go to the T state, my whole conformation of that subunit changes. I tell the other 3 subunits I'm connected to, hey, I'm in the T state right now. You should probably also be in the T state. And so they change, they release oxygen, cooperativity. If we're always in the R state together and we never change, then there's nothing to talk about, right? You're just always in the R state, so there's not going to be cooperativity anymore. If I bind oxygen, what am I going to tell the other 3 subunits? I'm already telling them, hey, I'm ready to bind oxygen. So I have no message to tell them. If I release that oxygen, well, I can't go in the T state. It's impossible. So I have no information to relate to them. I can't tell them, hey, I'm in the T state now because I release oxygen. They still get the message of, let's bind oxygen. So cooperativity is lowered and is 1. So this is one of those good test questions that really test your understanding of, do you know what it means to be R and T state? Do you know what R and T state means in terms of oxygen affinity? Do you know what the Bohr effect is and how that relates to T and R state? Do you know what a Hill coefficient is and how the R and T configurations influence that? So a question like this, don't be shocked if you see something similar next week, right? This level of thinking is really what I want out of you as upper degree students, upper level students, to start to train your brains to make these connections of taking the base information that I told you and applying it to these concepts. All right, so any questions about anything we have covered up to this point? All right. So in this case, the Bohr effect is non-existent because there's no T state. Yes, the Bohr effect only works because under low pH, you favor the T state. So you are forcing that transition from the R to T state. If you are a mutated hemoglobin and have no T state, then there's nothing to change, right? So you can be in acidic conditions, you're still going to be in the R state. So there's nothing forcing you to get that oxygen up. Unlike normal hemoglobin, you go to that area of low pH, well, now you're going to want to be in the T state, you release that oxygen. So yeah, that Bohr effect, the effect of the Bohr effect is lowered, or I would say there is no Bohr effect with that version of hemoglobin, which means you're probably not too good at exercising because your muscles don't get that continuous supply of oxygen. And so you probably get tired super easily after exertion with that form of hemoglobin. All right, so this question, let's see what the time is. I think I'm gonna skip this question. This just goes with free diving, and basically, I'll just answer it, I guess, instead of just skipping it. But this question is, people who do free diving, that is diving very, very far underwater without any like gear, one of the things that they will do before they free dive is hyperventilate. So they'll breathe very fast because they think that if you breathe very fast, you breathe in more oxygen. And does this actually work based on hemoglobin? And it doesn't. It actually lowers the oxygen concentration because when you breathe very heavily like that, you're increasing the amount of CO2 inside your body. And you don't get rid of CO2 easily when making shallow breaths. If you're breathing super fast like that, you don't give CO2 time to really get into your lungs. And so your blood becomes more acidic. If your blood becomes more acidic, the hemoglobin inside your blood, because of the Bohr effect, will be in the T state. And so before you actually dive underwater, you're going to release the oxygen already, which actually decreases your ability to dive further underwater because you don't have that hemoglobin around that has oxygen stored. You already released it while hyperventilating. Another part of this, when you are suffocating and you feel like, oh, I need to breathe oxygen, a fun fact, there's no way for your body to know how much oxygen you have in your blood at any given time. We don't have any oxygen sensors. We do have carbon dioxide sensors. That has to do with blood pH again, right? So if your blood pH goes really low, that means you have a lot of carbon dioxide. which means you have not breathed out in a long time, which means you need to take a breath. So when you feel like I really need to breathe, it's not I need to breathe in. It's more like I need to breathe out. I need to get all this carbon dioxide out of my body. And in doing so, you'll take in oxygen to replenish stuff. So that's just some fun facts there about hemoglobin and about breathing and about hyperventilating and free divers as well. Let's see where we're at. All right. Yeah, because for time's sake, we're going to skip the MWC model, and we're also going to skip the KNC models of ligand binding. So these two models, they go over like how exactly cooperativity works. We don't actually know how it works, but these two models would go over like, this is what we think. Just for time, I'm going to skip going over those because there are more important things I want to get to. And briefly, let's talk about sickle cell anemia. And what sickle cell anemia is that your red blood cells become sickle-like, and they can get stuck in your veins, your capillaries rather, and cause a blood clot. And this can be very dangerous. However, if you're a heterozygote for the sickle cell red blood cells, this is good because it protects you from diseases, right? Malaria. The reason why sickle cell happens is due to one mutation in hemoglobin. In hemoglobin, if an aspartic acid at position 6 is mutated to a valine in position 6 at your beta chain of hemoglobin, what will happen is that you'll make a new hydrophobic interaction, which is being shown in these images. And your hemoglobins will start to chain together. That's what's showing here. Normally, your hemoglobins don't interact with each other, right? You have 4 cell units coming together and make one protein. These proteins don't interact with other hemoglobins. If you have this mutation, they will start to stick together and make these long protein strings. And these things are sharp, and they will bust through your red blood cells and also cause this sickle formation as you just have long strings of hemoglobin. So that's what sickle cell anemia, that's how it happens. You have exactly one amino acid mutation causing your proteins to stick together. This is treated with the drug hydroxyurea. And what hydroxyurea does is it helps to stimulate your body to make fetal hemoglobin. Remember, I talked about that already today. And fetal hemoglobin does not have this interaction. Fetal hemoglobin, even with this mutation, does not stick together. They have slightly different chemistry. And so you do not get these sickle cells with this drug. So that's a way we can treat sickle cell anemia. And that ends our discussion on hemoglobin. So are there any questions whatsoever about any of the things we talked about with hemoglobin, myoglobin, cooperativity, RT state, Bohr effect, Hill constants, anything at all that you're still not sure of, you want me to go over again, now's your time to ask. And while you're thinking about that, let me just, I'm just going to fast forward to see where we're at. All right. Okay, so we are not going to get to go over 7.2 today. We don't have enough time. So what I'm going to say is that 7.2 muscle contractions, that will be on test 3. Everything about hemoglobin and myoglobin before this point will be on test 2, right? So before this, test 2. From this slide onwards, test 3. And since we only have like 5 minutes, I'm just gonna say we'll call it here. On Monday, we're gonna pick up at 7.2 and talk about muscle contractions and continue on. But yeah, we have now covered all the material that will be on our test 2. I will put a homework assignment up to give you some practice on what we talked about today. If you have any questions whatsoever, email me, set up a meeting, pop by 12 o'clock today. I'll be around. By the way, if for some reason, like you ever come to one of my office hours when I say that I'm going to be there in my Zoom windows and it says I'm not there, feel free to send me an email. Sometimes I might be like on campus. That's been happening a lot now, where I'm not in my office, but I can be in a minute. So just shoot me an email and I'll pop on ASAP. But otherwise, hopefully you learned something interesting today. Hopefully your head doesn't hurt too much. Just look over these topics. I know it seems like a lot, but I think I'm confident everyone here can get it, taking some time to think about it. With that, hope you stay dry. Seems like those of us in San Antonio are expecting more rain today. So keep dry. Have a good weekend. And I will see everybody Monday. Take care, everybody.